Hi friends, today we are going to discussing about one important concept about DNA, that is why the ends of DNA stands represented as 5 prime and 3 prime instead of 5 and 3. As you all know, DNA is the genetic material for all living organisms starting from bacteria to higher organisms like humans. Virus is not a living organism. It is semi-living organism and is the connecting link between living and non-living organisms. So virus is not considered as a living organism. So starting from bacteria to higher organisms, DNA is the genetic material. As per the Watson-Crick model of DNA is a double helix in nature and it contains two polynucleotide strands which are antiparallelly oriented. This is DNA structure. It contains two polynucleotide strands and these two polynucleotide strands are antiparallelly oriented and each polynucleotide contains nucleotides. That means nucleotides are the basic units for DNA or polynucleotide strands. And these nucleotides covalently attached to each other are polymerized by phosphodiester bond. That means each polynucleotide contains number of nucleotides and each nucleotide connect to each other by phosphodiester bond. Each nucleotide contains three components. If you take the basic unit of polynucleotides and our DNA, that is nucleotide, each nucleotide again contains three components. One major component is nitrogen bases. For example, if you take DNA, it contains adenine, guanine, cytosine, and thymine as nitrogen bases. Or in case of RNA, the four nitrogen bases present in RNA are adenine, guanine, cytosine, and uracil. So the first component in nucleotides are nitrogen bases. Generally, these nitrogen bases are categorized into two types. Those are purines and pyrimidines. Purines are adenine and guanine. It contains one six-member ring and one five-member ring. So adenine and guanines are purine. These are common both in DNA and RNA. Whereas pyrimidines are three types. They contain only one six-membered ring. They are cytosine, uracil, and thymine. If you take the pyrimidines present in DNA are cytosine and thymine. Whereas in case of RNA, the pyrimidines present are cytosine and uracil. So these are the nitrogen bases. In nitrogen bases, there are several positions are there. For example, if you take purine, one six-member ring and one five-member ring is present. Whereas pyrimidines, only six-member ring is present. So the numbering given to the purines and pyrimidines are 1, 2, 3, 4, 5, 6. Like that they are given. If you see in purines, there are six positions are present. Whereas in pyrimidines, only six positions are present. And the numbering are 1, 2, 3, 4, 5. Like that they are given for purine and pyrimidines. This is the first component. Nitrogen bases are the first components in the nucleotides. And the second component is pentose sugar, that is ribose. Pentose means five. Five carbons are present. In this also we have to give numbers. Already we have given 1, 2, 3, 4 numbering for the nitrogen bases. But for sugars, the same number if we give the 1, 2, 3, 4, 5 like that, if we give the numbering to the sugar, there may be a confusion or ambiguity. That's why the positions represented in the sugar represented as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Like that they have given. For nitrogen bases, 1, 2, 3 numbering given. For sugars, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime like that given. And the third one is phosphate or phosphoric acid. It is a tribasic acid. Because of this phosphate, the entire nucleotides are DNA is negative charge. Because it is tribasic acid, that means three negative charges are present. So the total negative charge of DNA or RNA or nucleic acid is mainly due to the presence of phosphoric acid. These are the three main components of the nucleotide. So each nucleotide contains one nitrogen base, one pentose sugar, and phosphate. So these three components collectively called as nucleotide. If you take nitrogen base and sugar, nitrogen bases, several positions are there, numbering given as 1, 2, 3, 4, 5, 6, 7, 8, 9 like that. Similarly, for sugars, The same number if you give, ambiguity causes. That's why we are giving 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. If you combine the nitrogen base with the sugar, that is called nucleoside. If nucleoside attached with the phosphate, that is called nucleotide. So if you take the structure of nucleotide, it is like this. So it contains nitrogen base, sugar, and phosphates. So in nitrogen bases, the numbering given as 1, 2, 3, 4, 5, whereas sugar numbering given as 1, 2, 3, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So if you take polynucleotide stand, it contain this is phosphate, this is sugar. This phosphate, sugar, phosphate, sugar, phosphate, sugar. That means the polynucleotide stand backbone formed by phosphate and sugar. And this is the fifth position or 5 prime position, and this is the 3 prime position. So whenever whatever the length of the polynucleotide stand, one end is 5 prime, that is fifth position, and the another end is third position, that is 3 prime end. So that's why here we are representing in DNA, we are representing ends based on sugar positions. So that's why we are giving 5 prime and 3 prime. Here, these are the nitrogen bases. Nitrogen bases are in core or middle. So there is no numbering representation. Generally, they are involved in the bonding, that means interactions, complementary base pair. Only backbone formed by phosphate and sugars, and the numbering we are representing here based on depending on sugar. That's why here 5 prime, here 3 prime. And another thing is why you are giving prime is in order to avoid confusion or ambiguity between the positions of nitrogen bases and sugars. So here we are representing the ends based on sugar positions. That's why we are giving 5 prime and 3 prime. This is the basis for giving representing DNA ends either 5 prime or 3 prime instead of 5 and 3.